we'll hand over now to Pat Trannell. Good morning, Pat, or good evening for you. How are you?
I'm doing great, thank you.
So tell us about Illinois. Is it still the cold season there, or are you coming out the other side of it?
We have spring is in the air, so I think we're through the worst, but we never know.
Excellent. So it's Monday evening. What time is it over there, Pat?
It is 6:30 p.m. right now.
Good one. So just tell us a little bit about who you work for and what you do and how long you've been researching resistant weeds.
Yeah, so I'm here at the Department of Crop Sciences in Urbana, Illinois. I've been here on the faculty at this university for about 17 years now. Prior to that, I spent a little bit of time at Monsanto Company, but spent most of my adult career here at the University of Illinois, doing research around weeds, of course, and in particular, my focus has been on investigating mechanisms of herbicide resistance.
Yep. And so this study you're going to tell us about today, I won't steal your thunder, but I've been telling the Australian agronomists and farmers a little bit about this in the recent extension period because it is such a good story to tell, and it's an important one because we have really drilled into the community about rotating herbicides for a long, long time. But what you're going to tell us today, I think, Pat, is that mixing herbicides is even more important. So yeah, I'll hand over to you, Pat, and let you get into it and tell us that story, and I'll interject with questions as we go.
All right, great, Peter. Well, thanks again for having me here. It's really an honor and a privilege to be able to share our story with you. And so I'm going to tell you a story about some research that we did, which led us to the conclusion that mixing is more important. And the neat part about this conclusion is that it was reached from real data from real farms. So it wasn't just an experiment we dreamed up in the lab or in the greenhouse and put out in the field, but actually something we did from real farms. But before I get to that story, I'm going to basically divide up my presentation here in three parts. I'm first going to introduce herbicide resistance as an evolutionary event. And I'm sure many of your listeners are aware that herbicide resistance is an evolutionary process, but I don't think we can overemphasize this because the importance of this is that because resistance plays by the rules of evolution, any strategy we implement to try to delay or mitigate resistance has to play by those same evolutionary rules. So I want to lay out this evolutionary context of resistance, and then I'll talk about our study and our results. And then I will return back to this evolutionary context and explain why I think our results make sense in light of this evolutionary background. So let's start with a definition of herbicide resistance. What is it? Well, there's multiple definitions out there, and I'm sure you guys all have your own definitions, but the one I like to use is what I show here on the screen, which is simply an increase in the frequency of resistance alleles in a weed population after exposure to herbicide selection. And again, this really emphasizes the evolutionary implications or the fact that it is an evolutionary event. It's a change in genetics of a population due to selection. And so we can just give a little cartoon here just to get everybody on the same page. Here we have this imaginary weed population where these are individual weeds here. They have genes. In this case, this weed, I'm showing the ALS gene, and it has different alleles, one from the mother, one from its parent. And most plants have 2 copies of the normal wild-type sensitive ALS allele. But occasionally, in a non-selected population, very rarely, there will be an individual weed that has this rare resistance allele. So then when you spray the herbicide, it kills off the sensitive plants, the resistant plant, because it has that resistant allele, it survives, it transmits its alleles to the next generation, and this is what it looks like. Here's the initial population. After spraying, you can see an increase in that resistance allele. And in fact, we can quantify that. In this particular cartoon, it went from a frequency of 0.05 to 0.25. And the point here I want to make is that this is a very mathematical process. There are certain equations we can plug in that will predict and model evolution of herbicide selection and the resulting increase in resistance alleles. How fast it happens and some of the details, some of the numbers that would punch into the equation are listed here below in terms of how effective is the herbicide, what is the genetics of the resistance trait, what is the biology of the weed. All of those things will influence the equation, but the equations we know, they're mathematical, we can punch in the numbers and we can make very actually very good predictions. And so here's just a standard graph of resistance evolution. I plotted on the y-axis. You can think of this as the percentage of the plants in the field that are resistant over time. And this is the general shape of the curve. Now I have it showing here in this graph that about year 7 it starts to take off, or you start seeing resistance. It may not always be year 7, it might be year 5 or year 9. Again, that if you go back to the previous slide, lots of factors can influence it. But this is the general shape. And when we look at this curve, we can divide it into 2 phases. There's this phase here where it looks like it's starting to recur, resistance is starting to occur in year 7, 8, 9, 10, it just takes off exponentially. And that phase we could describe as the management phase. Another term for that might be the reactive phase. You have resistance, you see it in your field, you're now starting to change your management to deal with that resistance problem. Prior to that phase, however, we could think of as the resistance mitigation phase. At this point, year 1, 2, 3, it's not apparent that you have resistance in your field, and so you're trying to prevent resistance from happening. This is how we think about resistance. But I really want to change your way of thinking about this curve, because I think there's a much more accurate way of thinking about it. And that's shown here. And so here's essentially the same data, but I've changed the y-axis and put it on a log scale. Okay? And so we can put in the same time frame of these different phases. Here's year 7, where we just started seeing resistance in the previous graph, and that's the same thing here. So at year 7, the resistance frequency is 1%. So if you're getting 99% control, you're pretty happy and you don't think you have resistance, and that's showing up in this curve. Prior to year 7, you're not seeing resistance. But if you look at this curve, what's happening? Year 1, year 2, resistance is increasing, and by the time you reach year 7, where are you on this curve? It's not the beginning of the problem. You're basically, this curve is played out, and so now you're at the end of the process essentially. And that's really how we need to think about resistance. And so we're not trying to prevent resistance early on. It's already there in the field. It's just at a frequency below what we can see. And so as we're dealing with our weeds in the field, we should always think that we are in the management phase. We have resistance in our field. We need to be preventing or trying to slow down the rate at which it's increasing. Okay? So that's a basic overview, and then when we get to this phase, as I say here, it's too late by the time you can see it. So you need to really be thinking about resistance, trying to manage and prevent resistance before you can see it. Once you see it, it's too late.
So Pat, just let me interrupt.
Sure.
Just quickly, firstly, someone has just let me know that your slides are coming through a little bit slowly, so
okay.
And they're not keeping up with the commentary, so you might just need to think about that. Just leave them up for a little while.
Okay.
But also, just a question about what you're talking about there. Just give us a little indication of the background gene frequency in an unselected population of some of the different herbicides. So, you know, ALS versus a glyphosate or something. What sort of percentage, or, you know, what's the background frequency of resistance?
So, unfortunately, we don't know. That's the short answer. We can make some guesses and some assumptions, and there's a little bit of data out there to suggest what those numbers might be, but for the most part we don't know. A common number that's thrown out, which is, and again, there's a little bit of data out there. Actually, probably some of the best data has been done by Chris Preston, I think, in terms of looking at that background resistance frequency. But a typical number that we throw out is about 1 in a million.
Yep.
And probably 1 in a million and less. It depends on the resistance. So ALS resistance, which is relatively easy for weeds, is probably 1 in a million. Something like glyphosate, that's maybe harder, that might be 1 in 10 million, maybe 1 in 100 million. But the short answer is we don't know. Those are best guesstimates.
But we do know that different herbicides have different background gene frequency.
That's correct. And the other thing I'll point out is that seems like a really low rate, 1 in 10 billion, who cares? That's such a small rate. But I've done some crunching just looking at numbers. If you start multiplying how many hectares you have of ground that you farm across Australia and how many weed seeds are in the seed bank, weeds win numbers games. There's lots of weeds out there. There's trillions and multiple trillions of weeds out there. So even if it's a 1 in a billion event, it's still going to happen quite frequently, given the fact that you have 10, 100, or 1,000-fold more orders of magnitude of that in terms of the seed bank.
Yeah, and I'm just a comment came through from Andrew Story, actually, who said that ALS resistance gene frequency frequency than that would be in ryegrass in Australia is 1 in 40,000, and I think he is referring to Chris Preston's work there. So yeah, it can be very common, or it can be 1 in millions, as you say, for other herbicides. Okay, thanks, Pat.
Okay, so that's just a backdrop of resistance, putting it in an evolutionary context. So I want to talk about our ryegrass. So I know you guys do with ryegrass. That's probably your #1 weed, at least in several parts of your area. Our #1 weed in much of the Midwest and much of Illinois is this species here, waterhemp, amaranth tuberculatus. It doesn't look like much here. This is growing in a cornfield. But what can happen, there's several biological attributes to this species. I don't have time to talk about all of them, but one of them is that it tends to emerge over multiple flushes during the growing season. And so you may have a pretty clean field midway through the growing season, but then you pull in with your combine and there you see some water hemp plants above the canopy that are setting seed. Water hemp is a prolific reproducer. A single plant can produce potentially 1 million seed. And this is one of my favorite photos here. The carcass here in the middle is a water hemp plant that was cut off with a combine, and this is just the remains, and then all this green is the progeny of that one water hemp plant. And this was actually a glyphosate-resistant site. So it just shows the incredible reproductive potential of the species. And so it may start out, you just have a few plants scattered here and there in your field, but then over time it builds up its population very quickly. And this is a soybean field, and most of what you see here in the foreground is all water hemp. So it can be a very aggressive weed. In this case, it's taken over the field. And sort of like your ryegrass, one of the characteristics of water hemp that makes it a very good weed is it's very good at evolving resistance. And so this is a timeline of the different types of resistances that water hemp have evolved. And so we're dealing with populations now that have 3, 4, and in some cases even 5 different herbicide resistances, resistance to 5 different herbicide modes of action in a single population. So this is the challenging weed for us. And so this brings me to the specific report or publication research project that we did. And I want to tell you a little bit about how this project originated. And so back in 2006, we first confirmed glyphosate-resistant water hemp in Illinois. And so at this location, this was a couple hours south of where I'm located here. As we drive back and forth, we did on-site research at this location for a couple of years following its identification. And as glyphosate resistance was starting to expand from the single field and show up in other fields, and as we were driving, to and from this research site and around the area, it was really remarkable how we would see in one field, it's pictured here in the upper left, a field that was a total mess, overrun by water hemp, presumably glyphosate-resistant water hemp. But then maybe right across the road from that, you would see a very clean field. And so this really begged the question, what's different between these two fields? Is there something the farmer is doing differently? Wouldn't it be great to know what is the single biggest difference between these two fields that we could use to understand why one field has glyphosate resistance and why the other field doesn't? And so that was really the origin of this study, is to try and ask this question, what is the factors that are related to the presence or absence of glyphosate resistance? And so we took this landscape scale approach, or an epidemiological approach, to understand this question. Typically when you have a research question in science, you will have a hypothesis, you will put out different treatments where you vary factors, and then you look at the results. Well, in this study we took a different approach. We already had the results. We had these fields that had or did not have glyphosate resistance, and so now we could ask the question, well, what were different about these fields? So we kind of went backwards. We had the result first, and then we asked what were the treatments that were imposed that got to those results. And so the way we did the study then is we went to a retail applicator at this location, a person who sold herbicides, who actually in some cases applied the herbicides on these different farmers' fields. And we basically said over about a 700 square kilometer area, we say, can you give us about 100 fields, or identify 100 fields for us, about half that have a glyphosate-resistant water hemp problem, about half that don't, and then we'll try and figure out what were the conditions that led to the resistance in those that had resistance, and what apparently was responsible for not causing resistance in the other fields. And so we identified those fields, and we also had management records, or he was able to provide to us management records of those fields. And so we looked at various things, how often glyphosate was used, how often herbicides were rotated, how many herbicides were applied per year, whether a pre-emergence herbicide was used, basically anything we could find. We also looked at characteristics of the weeds in the field. For example, we assumed that the more water hemp you had in the seed bank, the more likely you were to have glyphosate-resistant water hemp. So we tallied that information. We looked at characteristics of the soil. We also looked at characteristics of the landscape. And if I can just skip ahead here, why would we look at the landscape? Well, so here's an example of a field which is surrounded by trees. So we would hypothesize that water hemp is an outcross species, and so maybe if the field is surrounded by trees, that would form a barrier to pollen, and so that would sort of protect this field from outside sources of pollen, and maybe this field would have a less chance of being resistant. We also know that water hemp seeds move in water, partly why it's called water hemp. And so we hypothesized that if you were a field that had a water course that might seasonally flood, that would be a source of water hemp seeds from maybe your neighbors or from other fields, and so therefore you might be more likely to get resistance. So again, kind of just casting a large net, collecting every factor we could think of that may in some way influence the outcome of resistance. So we collected all that data. In addition, we collected seed, brought that back to the greenhouse, and screen it for resistance. And so in this bottom right photo here, I'm showing one field where there was a high level of glyphosate resistance, one field with a lower moderate level of resistance, and then over here is a field with no resistance. So we took this as our response variable, glyphosate resistance, and looked at all of these factors to see which one were associated with resistance. Okay, does that make sense? Any questions there, Peter?
No, it's all good, Pat. It makes perfect sense, yeah. And I think you're being a bit kind saying that water hemp is your ryegrass. I think we'd be horrified if ryegrass looked like that. Yeah. It's a very aggressive weed.
It is. It's good job security for me, though, so I kind of have a warm spot in my heart for water hemp.
Yeah, you're in a great area, yeah.
So, some of the results, and I'll go through this fairly quickly, but the first result I'll mention was that of all these factors we looked at, management, soil, landscape, weeds, management came out as being most associated. And that occurred regardless of whether we looked at the percent of the plants in the field that were resistant or simply looked at whether there was or was not glyphosate-resistant water hemp in the field. Management factors were most important. And I actually take this as a very good finding. This means that you as a farmer are in control. I was a little bit surprised by this finding, to be honest. I thought, you know, if you had that water course running through your field, or if you had a high water hemp seed bank density, you would probably be destined to get resistance regardless of what you did. These results said, no, that's not the case. What you do matters. And so, like I said, that's good news. That means you as a farmer are in control of the situation. If you do the right thing, you can delay resistance evolution.
Just one question there, Pat.
Sure.
Landscape is not that far behind, is it, in terms of its 0.44 instead of 0.53? So what aspect of landscape are you talking about there that might influence resistance?
So if we go back here, I mean, these are all the various factors. And we didn't really mine those a lot further to determine that. Part of it could be, I think the water is a big thing. We do know that water hemp moves through water, and so if you are, you know, situated close to fields that seasonally flood, and so there's new water hemp brought in, that could be a factor. Size of the field, I mean, all those factors, we didn't really pick that out further.
Okay.
Okay, so management factors, management is most important. What aspect of management? And so we did this card analysis, which is classification and regression trees. And I should have pointed out that I'm obviously not the only author on this study. There are several others and several very good statisticians who would probably love to explain the card analysis in much more detail than I would care to explain. All I'm going to say is the card analysis, it's basically a way to try to identify those factors that best explain the variance. And as we did this card analysis, it pointed to herbicide use factors as being most important. And so that just set the stage that we really need to look at these herbicide use factors and how do they come into play. But the problem with this cart analysis is a chicken and an egg problem, or a cart before the horse problem. In other words, it implicated herbicide use factors, but we didn't know were the management factors influencing resistance or because we had records over time, maybe as resistance was starting to show up, farmers were changing their management. So which was causing which? And so we had this problem. And so the way we address this problem is our records went back to 2004, and resistance, glyphosate resistance, as I mentioned, was confirmed at this location in 2006. So we thought, well, let's look at management practices in 2004 through 2006 before glyphosate resistance was known in the site, and ask the question, could we look at the management practices that were in place in 2004 and 2006 to predict whether or not there would be resistance in 2010. And so when we did that, the striking finding that came out was that herbicide mixing was very strongly associated with the practices that were, herbicide mixing that was in place in 2004 and through 2006 was very strongly associated with whether or not the farmer or that field would have resistance in 2010. And so these graphs are basically just showing different ways that we looked at. We looked at it in many different ways. The top here is the probability of resistance. The bottom is the proportion of the plants that were resistant. And again, the conclusion is the same regardless of how you look at it. Those farmers that were mixing herbicides, or those fields that were receiving mixes of herbicides in 2004 and 2006, were less likely to see resistance in 2010. And so just to kind of quantify that, if a farmer on average was using 2.5 herbicides per tank mix in 2004 through 2006, they were about 80% less likely to have glyphosate resistance in 2010.
So Pat, can you tell us a little bit more about that? We're talking about glyphosate tolerant crops here, aren't we? And so we're talking about some growers obviously who are just using glyphosate only in the glyphosate tolerant crops, but the guys that are mixing that 2 or 2.5 modes of action, what are some of the other sort of herbicides that they can use in those crops?
And so these are across all herbicide applications. So these could include pre-emergence applications where they're maybe mixing a triazine type herbicide with so apologize, you guys use different group numbers than we use, and so I can't refer to the group numbers that's meaningful to you, but things like metolachlor, I think you guys would be familiar with. So we'd be using like a metolachlor and an atrazine in a premix, maybe including a burn down herbicide in there as well, maybe some 2,4-D or something, and then coming back in season with maybe in soybeans they might be applying a glyphosate along with a PPO inhibitor like a diphenether. So you're right. I mean, and in corn we have even more options. And so, and in corn and soybeans, they could be using glyphosate in both of those crops. These modes of action were not glyphosate specific, so we didn't treat glyphosate special in these. And when we tally this information, it's just looking at how many modes of action were in that tank.
And was it necessarily a tank mix, or was it sometimes, you know, a pre-em followed by glyphosate in crop?
So some of these fields definitely received both of those. What we're showing and the way we analyzed the data in this was just looking at how many modes of action per tank per year.
And the growers, there's a question came through, are they typically mixing it at full rates, or?
So we'll talk about that a little bit later.
Okay.
We did not quantify that in this experiment or in our analysis of these data. We did not quantify. So we'll talk about what has to happen for that mix to be effective. And we did not quantify that simply because we couldn't, and we can talk about that a little bit later, okay?
And just to quantify one more thing, the herbicide rotation on the right there, that is, you know, using, say, glyphosate this year and using a different group of herbicide next year. We're talking from one year to the next there, aren't we?
We are talking from one year to the next, and again, we're not treating glyphosate special. We're just looking at, and those indices we use are fairly complex, but it's just looking at how diverse were the herbicide rotations from year to year.
Yeah.
Now, another thing I glossed over here is there are some other factors that were important. Another factor that was very important was how often they use glyphosate, as you would expect. I mean, if a grower is not using glyphosate at all, they're probably not going to have glyphosate resistance. So just, you know, the frequency of glyphosate use was an important factor as well. But that should be common sense, I think, to everyone, right?
Yeah.
Okay, and so as you mentioned here on the right, you were talking about herbicide rotation. Basically, this is a pretty flat line. If anything, it was the opposite way we had expected, but I would just say it's a flat line because it's a pretty small slope and there's a lot of noise there you can see. But the point there is that rotating herbicides, whether or not the grower was rotating herbicides in 2004, 2006, basically did not predict whether or not they would have resistance in 2010. So these are the facts. These are the observations. I mean, this is sort of, we took all the data, we took the glyphosate resistance status in 2010, we took all the factors. These are the results we got. So now the question is, well, why did we get these results? And so now I want to return back to sort of taking this evolutionary perspective and using evolutionary theory to explain why we got the results that we did. And so why is herbicide rotation not particularly effective? And I should back up and say, we're not the first people to conclude that herbicide mixing is better than herbicide rotation. I think, as I said at the beginning, what's nice about our study is it came from real fields managed by real farmers, okay? And we came to this conclusion in a real world setting. So now, again, how do we explain these results based on the evolutionary underpinnings of resistance evolution? And so the reason why herbicide rotation is not particularly effective is because that in order to understand why it should be effective, you have to understand this idea of a fitness cost. So the basis for herbicide rotation to be an effective herbicide mitigation strategy is there's a cost of resistance, and this cost is a fitness cost in the absence of that herbicide. Okay, so what do I mean by this fitness cost? Well, a plant that is resistant to herbicide A is obviously at an advantage relative to its neighbor that doesn't have resistance to herbicide A when herbicide A is applied, right? The resistant plant will survive, its neighbor won't, okay? Well, it was generally assumed and actually demonstrated for some of for our very first case of herbicide resistance that a plant with resistance to herbicide A is actually at a disadvantage if herbicide A is not applied. And so if that's the case, and we thought that initially, we scientists thought that would be the case, then herbicide rotation makes a lot of sense. On the other hand, unfortunately, however, it turns out that most of our herbicide resistance traits have very little fitness cost, and actually they have basically a negligible fitness cost. So the whole premise for using herbicide rotation doesn't really work for most of our herbicide resistance traits. Now I can demonstrate that to you graphically, or I'll try to demonstrate that to you. So this is the graph I showed you before. And so this is showing herbicide resistance evolution to herbicide A if herbicide A is used continuously, okay? Now here I'm showing resistance to herbicide A. So this is resistance percentage. I should have noted this is resistance to herbicide A when this herbicide A is used in rotation with herbicide B. And so it looks like it's delaying resistance, and it is to an extent. But what we have to think about is we're only using herbicide A every other year. And so here I've highlighted the use of herbicide A in brown. And so we can compare like number of applications of herbicide A. And so here's after 2 years of using herbicide A continuously versus 2 years in rotation. And then in theory, when we rotate away, because there's no fitness cost, that resistance frequency doesn't go down. It just stays the same. And then we come back and use herbicide A again, it jumps up again. We rotate, it stays the same. We use it again, it jumps up again. So the point I want to make, if you look up here at this 8th, after 8 applications of using herbicide A continuously, we'd have 10% resistance under this scenario. If we use herbicide A in rotation every other year, after 8 applications, we still have 10% resistance. So this graph really shows this idea that by rotating herbicides, you buy time because you can use it for more years, but you're still only getting to use herbicide A 8 times and you're at the same frequency. So herbicide rotation buys time, but it doesn't buy you applications. Whether you use herbicide A continuously or in rotation, you can use it about 8 times if there's no fitness cost. Now, if our herbicide resistances did have a fitness cost, this blue curve shows you what it would be like, and this shows the effectiveness of herbicide rotation when you have a fitness cost. So here you use the herbicide A, it increases, you rotate away because there's a fitness cost, that fitness penalty works in your advantage and drives down the resistance. You use herbicide A, it comes up again, and then it gets driven down a little bit by that fitness cost. And so we can plot the like number of applications again, and you can see here now, if you used it 8 times in continuously, you'd have 10% resistance, but in rotation, if there is a fitness cost, it's quite a ways down there yet. So in this scenario, which is actually being pretty generous in terms of the fitness penalty, it's buying you about 3 applications. So even if there is a herbicide fitness, or even if there's a fitness cost to resistance, it's buying you a few applications. Not a lot of applications, but it does buy you some. More importantly, it's buying time.
If we can just talk about fitness costs a little bit more, this is great stuff. You mentioned atrazine. We know there's a fitness cost for atrazine. Can you think of any other herbicide resistance examples where there's a fitness cost?
So target site, there's different types of atrazine resistance. Some is due to target site, some is due to metabolism. Target site atrazine has a pretty substantial fitness penalty, probably about 30%. Those plants grow about 30% slower. They produce about 30% less seed. That is a big fitness cost. No other resistances, maybe growth regulator 2,4-D resistance. Some of those have fairly substantial fitness costs. Some of them maybe do not. Some of the early 2,4-D growth regulator type resistances did. Beyond that, most of our fitness costs, when people try to measure it, a lot of times they can't even measure it. It's so small they can't measure a difference.
Yeah, the only other one I'm aware of is Martin Villa-Ayoub, who did a lot of research at RE, measured a 30% fitness cost for one of the target site mutations for clethodim resistance in ryegrass, so the 2078 mutation, but other mutations for clethodim had no fitness cost, so it was a bit academic really. But also, so there was a question came in, that's the herbicide. What about different weeds? Do different weeds have different levels of fitness costs? Say for atrazine, do some weed species have more fitness costs than others?
Yeah, and so I generalize an awful lot. So for example, most cases of ALS resistance show no fitness cost. There's a published study, which I think is a very good published study by Francois Tardif from Canada, who showed that an ALS mutation in a pigweed, I don't remember if it was red root pigweed or smooth pigweed off the top of my head, but in that particular pigweed, a particular mutation had a high fitness cost. Other people have looked at that same mutation in other species and they have not seen a fitness cost. And so there is some variability among species. The other thing that you have to keep in mind is that initially when a plant has this mutation that causes a fitness cost, there are other mutations that can occur in that plant that can overcome or compensate for that fitness cost. And so particularly where you have very genetically diverse outcrossing species like loliums and waterhemp, initially maybe there is a fitness cost, but give it a little bit of time and it will evolve to overcome that fitness cost. So that's something else to keep in mind.
The other thing about fitness costs is you spray the herbicide and all the other weeds die, and then we just have the resistant weed left. So it may be less fit, but you've taken away a heap of its competition, and so it can grow quite well.
You have, but there also are probably, and it depends on the soil seed bank, but there are probably some sensitive individuals of that same species in the soil seed bank that will come back the following year.
Yeah. Now, there's some other questions have come through. One of them here is all focus of the modelling so far has been on the development of resistance to glyphosate. Have you modelled the impact of rotation versus multiple mode of action use on development of resistance to other modes of action?
Well, you know, that's where the models break down, are limited to some extent because it depends on the types of mechanisms of resistance that you can have. To be honest, this model here is not really herbicide specific. This is sort of a generic model that we're looking at right now. But those models, yeah, go ahead.
Oh, so you could say yes, really, but this is sort of conceptual, really, isn't it, rather than any particular measurement of the fitness cost of weeds.
That's correct. This fitness cost that I'm showing here in this blue curve, I believe I put that number at I think it was 25 or 50% fitness cost. I don't remember. So pretty high. That's about as high as we're going to see in most of our weeds. And that's the other point. You know, we can quibble over whether it's a 5% fitness cost or 10% or 15%. The point is, even with a high fitness penalty that I'm showing here, you're only gaining about 3 applications. Okay.
Yeah.
So even with a high fitness cost, in one farmer's lifetime, that doesn't help that much. When, you know, over
Yeah.
multiple lifetimes, you can drive down that resistance. The point is, no matter how much fitness penalty there is for in the absence of the herbicide, the fitness advantage when the herbicide is applied is so much greater.
Yep.
So the slope of the line going up is always going to exceed the slope of the line going down.
Yeah. Just one more comment before we let you keep going. Another one came through that I think Chris Preston measured some fitness cost in one of the glyphosate resistance target sites in ryegrass, but I guess the others had no fitness cost, so we end up selecting for the ones with no fitness cost.
Yeah, that's correct. And you know, determining fitness costs is not trivial. It's very difficult to do. There's lots of studies on fitness costs, and sometimes the studies reach different conclusions. And I'm not by any sense saying that that particular study by Chris Preston was wrong, but there are lots of conflicting studies. It's very hard to do these studies because you have to, you know, for reasons that we don't have time to get into, I'll just leave it at that. They're very difficult to do that, and we can get different results ranging from fairly high fitness costs to non-detectable. But again, I just want to come back to this point. Whether it's 10% or 20%, it doesn't matter that much. It's going to buy you a few applications, but it's certainly not going to save that herbicide or prevent resistance from occurring to that herbicide. Okay?
Yeah, that's great. Yeah, we better keep moving.
Okay, so we're almost to the end here. So just now to wrap this up, why do we think from an evolutionary perspective is herbicide mixing effective? And this is a little more easier to explain, I think. And it simply comes down to the fact that the probability of a plant being resistant to two herbicides is the product of the probabilities of being resistant to each of the herbicide. And so you asked this question at the onset about what is this initial frequency of resistance? Well, if the initial frequency of resistance is about 1 in a million to herbicide A, and it's similar 1 in a million to herbicide B, the probability that a single plant will be simultaneously resistant to both herbicide A and B is equal to that product. So in this case, 10 to the minus 12th, or 1 in a trillion. So you greatly decrease the chance that a plant is going to survive both herbicides and transmit its alleles to the next generation.
I guess we need to stress that that's before herbicide selection, isn't it?
Yeah, this is assuming that you're going into a field where the resistance frequency of both is low. Okay? And so that brings me to this next slide. And so there's lots of caveats to this idea of why mixing is going to be effective. The first one I'll point out here on the slide is that in order for this to work, you have to have a different resistance mechanism being required for each herbicide. So if you have two herbicides that are both ALS inhibitors, they both have ALS as the target site, and a single mutation can simultaneously confer resistance to both of those herbicides, then that's not going to be, it's not going to be 10 to the minus 6 times 10 to the minus 6. It's going to be simply 10 to the minus 6. Similarly, if the herbicides can be metabolized by the same enzyme, a single mechanism could overcome it, and so those two herbicides are not going to be an effective tank mix in terms of resistance mitigation strategy. Now, fortunately, we know the sites of action of our herbicides, and so we can make our selections based on herbicides with different sites of action. But unfortunately, we don't know the different enzymes that can metabolize our different herbicides. So the second point there, or the second sub-bullet, we're kind of left to have to guess at. The next big caveat is that the two herbicides that are used in the mix, they must be effective alone, and they must be targeting the same individuals. Okay, so some examples of what I mean by that, or what doesn't constitute that or meet these criteria. If you're mixing a foliar active herbicide with a residual soil applied herbicide, those two herbicides are targeting different individuals on that field, and so that's not going to be an effective tank mix from a resistance management perspective. If the two herbicides aren't alone able to kill the weed, okay, because they're being applied at a sublethal dose, then there again you're not having an effective tank mix combination for a resistance management standpoint. I like to think of it as hammers, okay? Your herbicides are hammers, and each hammer alone has to kill that weed. If you're not having each hammer alone killing the weed, then it's not going to be an effective tank mix strategy. Okay, you mentioned just previously, Peter, that this 10 to the minus 6 assumes a non-selected population. And that's this other caveat here, is if you already have a significant resistance population in that field, then mixing those two herbicides is not going to be effective from a resistance management strategy. Talking about pre-emergence herbicides, ideally, again, you want those herbicides to be targeting the same individuals. So if you mix two pre's, one which has a very short soil residual and the other has a very long soil residual, well, that one with the long soil residual is going to be targeting weeds by itself. And so you're not going to have two herbicides working together. And then, of course, the final point I'll make, which I think you guys drill into your farmers are quite well, is even the perfect mix is not bulletproof. In other words, herbicides themselves are not bulletproof, and we've just been talking about herbicide management. That's one part of weed management. Ultimately, your weed management has to be more than just herbicides. It has to be lots of other diverse strategies as well. So I think having these caveats up on the slide, this is my last slide. I probably will raise a lot of questions, a lot of points to clarify, but really, you know, the take-home message is that the theory behind why mixing works, I think, is pretty obvious if you understand evolution, and you have to keep in mind these caveats. The idea is that each of these herbicides alone is killing the weed, and therefore you're reducing the likelihood that resistance alleles will be transmitted to the next generation.
Yeah, that's great, Pat. And look, you've got a couple of really good points on the slide there that probably things I'd overlooked a little bit, particularly around the business of having herbicides, you know, a soil herbicide versus a, you know, a post-emergent foliar activity herbicide. So it'd be good to dig into that a little bit, because in my mind, I would have thought that it would have still been a good idea to use perhaps a good, say for ryegrass, a good soil residual pre-emergent before sowing Roundup Ready canola, and then using the Roundup in crop, and then I would have called that an effective mix. But would you say that I've got that wrong?
Well, as long as the foliar applied herbicide is going to be targeted effectively on those weeds that are escaping the residual. So one of the issues we get into, and you know, you guys do the double knock strategy, and a question I get asked a lot is herbicide mixing the same as making two applications in the same season? And for the most part, I think the answer to that can be yes. So whether the herbicides are applied at the same time or sequentially within the season is okay. It's okay to do it sequentially as long as that, so in this specific example, that foliar application is coming back timely when those first weeds that escape the soil applied herbicide are still small enough to be effectively controlled by that foliar herbicide.
Yeah, okay, so we are in that situation a fair bit, so that's good. And also your other point there about pre's having similar soil persistence and mobility, that's another important one. And so for us, trifluralin plus one of the other preems, whether it be propyzamide or boxagold or sakura, is the sort of obvious mix that we're talking about. And yes, there definitely would be differences in their length of persistence, so that's probably something else that's quite important here, I imagine.
Yeah, we want, you know, both of those herbicides working together. Again, that double hammer, that 1-2 punch. You want both of them hitting that every individual weed, ideally. And you know, Treflan doesn't have a lot of doesn't have a lot of mobility in the soil. It's probably that's the other thing to think about. If one herbicide is leaching through that soil fairly quickly and getting lower in the seed bank, where it's not targeting the same individuals or those weeds that are emerging from very shallow in the soil, both herbicides aren't having a chance to kill that weed. That again, it comes back to that. You want both herbicides, or all three herbicides, or however many you have in the mix, all having a chance to kill that weed. If only one is killing the weed, now you're back to selecting that weed with a single mode of action, a single herbicide.
Yeah. Okay, everyone, make sure you put some questions in now, because this will be your last chance to ask questions of Pat directly. Pat, someone's just put up on Twitter, just keep upping the rates. You know, that's the sort of, they've put that up as the answer. I'm just wondering, you know, let's talk about that for a minute. Is increasing rates, I mean, it's certainly not an alternative to mixing two products, but the issue of rate, and as we get resistance, does upping the rate help, or is it too late?
Well, some of our resistance mechanisms will overcome any practical herbicide use rate you will apply. So just upping the rate is not going to be an answer for some of our resistance mechanisms. Having said that, I'm certainly not promoting using low rates. You want to start out using high rates. You want to, going back, I have a point here on the slide. You can't obtain a lethal dose by combining 2 sublethal doses of the individual herbicides. You can do that, but it's not going to be an effective way from a resistance management strategy. You want to make sure you're using a lethal dose of each herbicide. And so, farmers might think, well, okay, I'll mix 2 herbicides, but I'm still only going to spend $20 a hectare or whatever you guys spend, and so I'm going to cut the rate of both. That's not where we want farmers to be going. We want them to be using lethal doses of every herbicide they're putting in that mix. And I know that's a challenge for, labeling in terms of economics is probably the biggest challenge. But I guess I tell farmers you either pay now or you pay later.
Yeah. So Pat, there's some other questions here. One is how many herbicides can you mix before getting antagonism between them, even compatible ones? And someone else put up a comment earlier that in Australia we like to do the witch's cauldron thing where we put in 3 or 4 products. So are we going to get to the point where we mix too many and cause antagonism, and then we're not getting an effective kill from each herbicide?
Yeah, I don't think it's really so much a question of how many, it's which herbicides you mix. Certainly some herbicides are better, some herbicides play better together than others do. And so it's not really so much the number of herbicides, it's more of that's where you need your local people that are looking at these different herbicide combinations to be giving you, look to them for advice about which herbicides play nice together and which ones do not.
Yeah, I guess as we put more and more in the tank, it becomes a bit of a 100 hectare tank mix experiment sort of thing, and it can go over thousands of hectares. So, I guess the answer is plenty of research in tank mixes. And the question of synergy has just come up, Pat. So, is there any examples of synergy that can be created by some combinations that spring to mind?
Yeah, so I really get careful when I talk about synergy and antagonism. So a good example of synergy that we have here in Illinois, and I don't know if this applies to you guys, but our HPPD inhibiting chemistry. I don't know if you have it. Is that familiar?
Yeah, we have that. Yeah, and just on that, you showed that waterhemp's evolved resistance to that as well. So that's velocity for us over here, or Precept. It's our go-to product for herbicide-resistant wild radish, and it's really holding the show together at the moment.
Okay, and so that with triazine herbicide shows some really nice synergy.
Yeah, and for us it's bromoxynil, and
okay.
I think I've got a feeling it's registered synergy. Someone might help me out in the questions panel there, but certainly there is discussions about synergy between bromoxynil and pyrosulfatol.
And so that synergy is good from a herbicide standpoint, but actually, and this will take a while to wrap your head around. We don't have time to explain this, but from a resistance management, synergy is actually not necessarily a good thing. It's better to have antagonism, assuming both herbicides are lethal to the weed. If they're antagonistic, it's actually going to be better from a resistance management. And the way to think about that is if you have 2 herbicides that are antagonizing each other, if the plan is, that means those 2 herbicides are less effective alone. They're less effective in combination than you would expect to be based on their activity alone. What that means is if the plant evolves resistance to one herbicide, then the antagonism won't occur, and so it'll be more easily killed by the other herbicide. So it's kind of a negative cross-resistance sort of idea. And so we have to
get my head around that a little bit. But
yeah, that's something.
Synergy is not sort of a holy grail for resistance management, is what you're saying.
No, in fact, synergy is nice for weed management, but it's not necessarily a good thing for resistance management.
Yeah. And another question that's come up, which is should we be using the double knock every time we use glyphosate? So the glyphosate followed by paraquat double knock. I'll let you have a swing at it, then I'll have one as well, Pat.
Yeah, I think the double knock is a really good way to go. If you have a weed that escapes, that's the one that's most likely to be resistant, and so you need to do whatever you can to kill that weed. we don't really do the double knock strategy here. I tell farmers, if at the end of the season or near the end of the season, if you sprayed your herbicides and you have weeds that are still alive, go out there, walk through those fields and get rid of those survivors. Obviously, that's not always practical, of course, but those are the ones that are going to contribute resistance. Those are the ones you need to target, and that's basically what that double knock strategy is doing. That's my feeling.
And I guess
I'll be interested in your opinion.
Well, in the ideal world, yes, we would use a double knock every time, but there's the real world which sort of says sometimes we get rain and other times we don't, and sometimes we have a big germination of weeds and other times we don't. So I think the combination of dry seeding, double knock, and then using more paraquat in those years when we have very small weeds as a single knock of paraquat is something that we often overlook. We often just revert back to using glyphosate because it's what we know. But on very small weeds, paraquat can be very effective. But I guess the other comment is everything we've talked about here is just about double knocks of all sorts. So really that's resistant weed management, isn't it? How many knocks can we get at the weeds of both chemical and non-chemical through the season that give us complete control at the end? And so the double knock is truly the answer, and there's more than one type of double knock. Yeah, so Pat, I mean, that's it for the questions that have come through, but just finally, can you just give us a bit of a snapshot of what's happening in your part of the world? We hear a lot about these glyphosate-resistant weeds bursting out of the top of crops that set millions of seeds, and how are farmers going? A few years on, it was 3 years since we heard all about it at the big conference in Fremantle here in Western Australia. How are they going now managing glyphosate-resistant amaranth weeds in the US?
Well, I would say twofold. One is we've definitely had an increase in pre-emergence herbicides. We've gotten away from pre-emergence herbicides in several of our crops, soybeans here in Illinois, for example, but that's really kind of come full circle. Pre-emergence are a big part of weed management. And then the other component is they're adding something in the tank with that glyphosate. And the PPO inhibitors have been used a lot. Unfortunately, we're getting a lot of resistance to those. And so there's kind of a lot of farmers are looking forward to, we have some new technology coming along, not new herbicide modes of action, but new herbicide resistant crops with 2,4-D and dicamba tolerant crops, and so those are coming along in the future. We're not quite sure when. A lot of farmers are kind of holding out hope for those. We think those will be a new valuable tool, but they're not going to be our savior, I don't think. So yeah, it's kind of sitting on the edge of the pants to see what's going to happen here over the next couple of years, because we are, as you know, we are starting to run out of options for some of these amaranthus species.
When I read a lot of the media coming out of the US, I hear what you've just said is lots of pre-em herbicides and tank mixes with glyphosate, and hopefully these new stack traits so we can use other herbicides. But I don't hear a lot about non-herbicide tools. Like, we're big into harvest weed seed control here, but I don't hear a lot about it from the US. Is that something people are starting to talk about, or are they still really focused on the herbicide options?
Well, we're talking about it. In fact, we actually here at Illinois, we have a Harrington seed destructor that we are starting to look at. One of my co-authors, Adam Davis, is doing some preliminary work on that. So yeah, there are, the scientists are looking at those. Again, the farmers, it's tough to get a farmer to think about anything other than spraying. We have as you guys have, large farmers farming large acreage. They like simplicity. And anything other than herbicides is challenging from a management standpoint, from a time standpoint, and so farmers are very, very reluctant to do anything other than chemical. We have a very corn, our system here in Illinois is corn and soybeans. It would be nice to rotate more with perennial crops like a forage, a legume, but it's just tough with our current corn soybean paradigm that we have.
Yep. So there is one last question that's popped up, Pat, and we might make this the last one, everyone. There's a question saying, have you looked into adjuvant quality in assessing the efficacy of herbicides? So have adjuvants got anything to do with it? Or I guess the answer probably is once you've got resistance, it doesn't really matter what adjuvant you put in there, it's still going to be resistant.
Yeah, that's true. I mean, adjuvants are important when you start, going back to the questions you were asking about synergy, antagonism, how many herbicides can you mix together? Sometimes, an adjuvant that's good for one herbicide is bad for the other herbicide in the tank mix. And so that becomes a challenge when you're trying to get, effective herbicides together in a tank mix, is trying to figure out an adjuvant that's compatible with both or all of them.
Okay, well thank you very much, Pat. It's been fantastic to see that presentation firsthand, and we really appreciate you taking the time out in the evening in the US to show us your research. I've really enjoyed it, and while I've read the paper and so on, I've learnt more today from you. So thank you very much, Pat, and thanks for everyone for tuning in. So thank you, Pat.
My pleasure.